the only thing that introductions do is make me feel old. So thanks for not doing that. So I realize as I look at the schedule this morning that I have 45 minutes and not an hour. So I'm going to be talking fast.
Like you usually do.
Okay. Disclosures, or as I call them, opportunities. I will note, however, just because I have to say it, that when I talk about triheptanone, you need to know that I have never taken a personal penny from Ultragenics. I don't consult for them. Again, I have no IP in the product, so everything I say is because I believe it. And I won't say much about it anyway today, because you're actually looking at the future. Thank you, Ronnie, for giving a shout out to Inform. I had the privilege of being able to start this group up about a dozen years ago now. Failure breeds success, and we didn't get it published, or funded rather, as an RDCRN site, and I said, what the heck, we're just going to do it anyway. And here we are 10 years later, and we have a meeting every year, and we get about 120 people who are coming, and so we've got 120 people who would know less about fatty acid oxidation disorders than we would have if we hadn't done it. We will be in Porto this year with the SSIM. The registration is still open, so please consider coming if you're going to be in Porto. It's a good way to spend a day and get some free food. As an introduction to this, I'm going to abbreviate this because I want to get to the details. We need to get more. Nobody's ever complained about me being too quiet. We want to. I want to give you a little bit of a 20,000-foot view of drug development, because that's what we're talking about here. Treatments in our disease have always been diet related, or the dreaded doctor, it hurts when I do this, then don't do that. So we're gonna move away from that and try to see how are we going to get better therapies, i.e. drugs, to treat our patients. And when you look at the drug development pipeline, traditionally you start with a whole lot of things, you have no idea what they do, and you're testing it to see if they affect something that suggests it might help you with whatever it is you want to treat. And that pipeline goes from hundreds of thousands of compounds down to one lead compound that then goes into clinical trials. And that process can take anywhere between 10 and 20 years. And one of the things that we can do in rare diseases is we have the benefit of not having a lot of people interested in our diseases, not having a lot of money to get into the field and sponsor clinical trials, but we have knowledge. We can start by saying, I can tell you what's wrong with this cell line or with this disease, and it manifests in this mouse model or in this cell line with absolute certainty. And if we can affect that biomarker, if you will, there's a really good chance that this is going to work. And so you can cut a lot of that preclinical work out and move straight to almost clinical trials with maybe just another 2-3 years of filling in the holes for the FDA. So we have an advantage over this undirected approach to identifying therapies. Now, if you stop and think about inborn errors of metabolism, which of course is what a fatty acid oxidation disorder is, you can think about two effects. And when I talk to my patients, I said, think about it as a block in a pipeline, right? You've got a problem if you've got a clogged pipe come into the sink in your bathroom. You've got two problems. One is you turn the sink on, or the faucet on, and nothing comes out. So in the case of fatty acid oxidation disorders, indirectly that leads to hypoglycemia. That's what's not coming out of the faucet. But you can also have a problem with what's backing up behind the block, and so instead of the water now in your kitchen sink, it's in your basement. Both can cause a problem, and the difference in the treatments that you can do depend on whether or not you're worried more about what you're not getting or what you're building up, as in something like PKU. And as a result, if you look at the where in the process you can affect the therapy of an inborn error of metabolism, you can go from this very classic and very simple substrate to product relationship and pretty much make it up from there, which in fact we did. This is a NAMA slide. There's American Metabolic Academy that I was talking about earlier. And what you can do is if the problem is too much of what's building up behind the pipeline, you can try to decrease what's going through the pathway. That's substrate manipulation in all of its forms. If you don't have what's coming out of it, that's substrate, that's product replacement in all of its forms. And for either of those, you can try to return the enzyme activity that's missing, whether you do that by an organ transplant or gene therapy or mRNA therapy. It doesn't really matter. It's all very simple when you think about it like this. And that's good because I'm simple. I don't like to get too complicated because it's too complicated. And so speaking of complicated, here's my stylized pathway of energy metabolism. And this is, I try to keep this as simple as possible, but I don't want to leave out details that you have to know about these disorders. Energy metabolism in the mitochondria is a pathway that requires is a function that requires three different pathways. Remember, fatty acid oxidation is a stress pathway, a stress process. You use it when you're not eating, so fasting. When you need more energy, you're sick, or you're out trying to run a marathon. And so for that to happen, you have to mobilize the fat from those places that, unfortunately, it often sits in places we don't want it. It is transported through the bloodstream to the cell where as a free fatty acid, where it needs carrier proteins to do that. It gets taken up into the cell, special carrier proteins to do that. And then has to be, if it's the standard long-chain fats that predominate in our diet and in our stored fats, it has to be activated to an acyl-CoA, transported into the mitochondria through the carnitine cycles, CPT1, CACT, and CPT2, where it's released as a free acylcarnitine again, and now undergoes the 4 steps of fatty acid oxidation. All right, so what about that is, why do we need all these other compounds that are listed up there? Well, if you'll notice, there is nowhere in fatty acid oxidation where there's an arrow drawn to glucose. This is like the first thing we say in NAMA to our trainees. If you think fatty acid oxidation is producing glucose, go home and come back next year. No, we just kindly point out the pathway. It's not there. What happens? Well, fatty acid oxidation makes reducing equivalents of electrons that through the respiratory chain are turned into ATP, which is used in gluconeogenesis now to make glucose. So it's an indirect effect, and you need the respiratory chain because you have 2 steps in fatty acid oxidation, the matrix step, part of fatty acid oxidation, the acyl-CoA dehydrogenases and the 3-hydroxy-acyl-CoA dehydrogenases that make electrons, reducing equivalents, that go into complex 3 and 1 respectively. And now am I talking too fast? Now you have some fuel coming out of those reactions. The 1 turn of fatty acid oxidation makes acetyl-CoA. That now goes into the TCA cycle, which in turn produces a whole bunch more reducing equivalents that fuel ATP production through the respiratory chain. And that's where your energy comes from. It turns out that about 1/3 of the energy from oxidizing a palmitate molecule, palmityl-CoA, completely comes from those first 2 steps, the first and the 3rd step of fatty acid oxidation, and everything else comes from the TCA cycle. So now you see why we need 3 cycles: fatty acid oxidation, the TCA cycle, and the respiratory chain. And I've already told you I'm an uncomplicated person, and I never believed that these things were all just floating around in the mitochondria, and one chemical found an enzyme that acted on it, and then that chemical went out and found the next enzyme. It's like these things have to be part of a complex where they all interact. And sure enough, after about 30 years, we were able to prove that. And there is an energy complex that includes long-chain fatty acid oxidation, the respiratory chain, and some steps of the TCA cycle. So it's all as a metabolite. So when all of these disorders first started being described, and I am old enough that I remember them all being described, we thought, well, what's the big problem? Long-chain fatty acid oxidation needs VLCAD and LCHAD and all those things, as well as the transporter molecules to get into the mitochondria. But medium-chain fats don't, and they would bypass all of that. All you got to do is give them medium-chain triglyceride oil, which is either pure C8, depending on what you're prescribing, or a combination of C8 and 10 and 12, depending on the product, and that should fix everything. Well, it didn't, and so the question was why? Back in very shortly after that was recognized, back in the late 90s, two very brilliant individuals, Charlie Rowe and Henri Grunengraber, got the idea and did some cellular studies that proved that the problem was TCA cycle imbalance. If you don't have, you already cut off half your reducing equivalents by starting with C16 and C instead of C, using C8 instead, and now you are relying on a reduced output from fatty acid oxidation. You've got the TCA cycle revved up to its maximum, and the whole process runs and it runs and it runs and it runs until. You see right here, acetyl going into the TCA cycle and making citrate until it runs out of succinate through succinyl-CoA. And why is that? Because you're making a ton of acetyl-CoA, but you're not making as nearly enough propionyl-CoA, which is the precursor for succinyl-CoA. And they came up with the idea of if we give C7 as a substrate instead of C8, maybe that will be a better fuel. Charlie managed to retire long before this ever got into clinical trials, and I was tasked with moving it forward. And to put a long story in a couple of words, it was finally approved back in 2020 as a treatment for long-chain fatty acid oxidation disorders. Now keep in mind that the symptoms for long-chain fatty acid oxidation disorders, you can break them up any way you want. But in large part, it's hypoglycemia, cardiomyopathy, and muscle disease, typically characterized by recurrent rhabdomyolysis. So as I said, probably now about 15 years after Charlie first had the idea for triheptanone, and about 10 years after I took it over, in collaboration with Ultragenics, we were able to get this approved by the FDA. Now, I'll just remind you that in this study, it was a relatively small study, which is typical of rare disease studies. It only was 28 patients, and so it just looked at every patient as their own control, how many episodes and what were their symptoms before they started triheptanone and after. And they were all on medium-chain triglyceride, C8, beforehand. So this was essentially a substitution therapy. And overall, the number of events were decreased by 50%. That was dominated by episodes of hypoglycemia, which were completely eliminated. It's vanishingly rare, and I actually don't think it's ever happened, that a patient on triheptanone gets honest-to-goodness, bona fide, documented hypoglycemia. It fixes the TCA cycle imbalance. It also reduced the heart symptoms, the cardiomyopathy, either worsening or development of, by about 75%. And the thing it worked the least well on was the muscle. It also improved the issue with hospitalizations. It reduced overall hospitalizations by 50%, and it decreased the duration of hospitalization by 50%. So the drug was approved, and these are actually now published studies. Long-term follow-up of a larger population, now overall about 120 patients in the first series of follow-up studies. It looks like it's even better than that. Those first 28 patients that we studied, we purposely looked for the worst-case scenarios. We wanted the patients who were bad enough that we would see an improvement. If you now look at it from all comers, it turns out that you still almost eliminate hypoglycemia. You probably decrease the cardiomyopathy episodes by something more like 90%. And you decrease the episodes of rhabdomyolysis, depending on how you calculate them, somewhere between more about 2/3. But there's still some unmet need there. The hospitalization picture gets even better. With a reduction instead of a 50%, more like 80 to 90% reduction. So this is good, but patients aren't asymptomatic, and when they are symptomatic, it's really life altering. Those episodes of rhabdo are really difficult for patients and their families, and the being in the hospital is not a good thing. So now let's go back to my original paradigm of making drugs for diseases. And I'll remind you of another basic tenet that you all learned back at this point, high school biology. That's the central dogma. DNA makes RNA, makes protein. And by looking at each of those steps, I can show you that we actually have drugs at all levels of the process that is inherent in the central dogma and that basic pipeline that I showed you previously from substrate to product. So for long-chain fatty acid oxidation disorders, I'll show you some data from my lab on a gene therapy for VLCAD deficiency and mRNA therapy for VLCAD deficiency. We've already talked about anaplerotic therapy, fixing the TCA cycle, and that's triheptanone, but not wanting to rest on a drug that's not 100% effective. We've been working on alternative anaplerotic agents. And then we also have drugs that are designed to hit some of the secondary problems that develop as a result of this multi-protein complex that I talked about. It turns out that LCHAD is not part of a trifunctional protein, but a quadrifunctional protein. No one's going to change the name just because we found another function, but it's involved in cardiolipin metabolism. And so now we're focused on trying to repurpose elimipritide, a drug that's being developed for respiratory chain deficiencies, and functions by stabilizing cardiolipins to see if that will help with fatty acid oxidation disorders. I will show you a little bit about a PPAR delta agonist, and I'll explain why that's important when I get there. And then chaperone therapy, both based on a function that we've called endogenous chaperone therapy, and also the more common exogenous chaperone therapy. Endogenous chaperone therapy is tricking the products of an enzyme to hang around longer than they otherwise should, or the substrate, depending on which direction you look at it. And there's nothing that functions better to help a misfolded protein or a misfolding protein to fold properly than a substrate analog. So the endogenous substrates if you can trap them in those binding pockets, suddenly you go from having no protein to more protein. And in the cases where that protein is functional, you can maybe get some benefit from it. So I'm going to talk to you about some alternative substrates for MCAD deficiency, just because all my patient meetings, everybody said, well, all you're doing is long chain. How come you don't do anything medium chain? So I'm going to talk to you a little bit about some stuff that we're doing for MCAD deficiency, including an alternate substrate, much as we developed for long chain fatty acid oxidation disorder. Oops, I hit the wrong button. There we go. So let's go back to the central dogma and intervene at the mRNA step. So before the pandemic, everything is in my head these days seems to be defined by pre-pandemic or post-pandemic. I was working with this little startup company called Moderna, who was making this stuff called mRNA. And they were looking for targets, and I managed to convince them to be interested in acyl-collating hydrogenases. What I'm showing here is that in control cell lines, there's very little. Let's go over here. There's very little VLCAD protein in null cell lines, and if you give them mRNA, you can make a lot of it. Here, showing a yellow signal is a lot of VLCAD going to where it's supposed to be in mitochondria. Here, showing enzyme activity going way up compared to the RNA and amount, and here looking at the protein signal. Does this help anything? The problem with mRNA therapy right now is it almost inevitably just goes to the liver. There aren't good capsids, there aren't good lipids that put it everywhere else. And so the question is, if you've got a disease where symptoms are liver, muscle, and heart-based, is getting the liver fix enough? Well, it turns out it's not perfect, but it actually has some effect. The VLCAD mouse model that's out there is not a really good model of human disease, because unlike humans, mouse use an alternative long-chain enzyme, the original one that was described back in the 80s when I was a fellow in Kei Tanaka's lab. We characterized LCAD deficiency and called that the thing that was going on in these long-chain deficient patients. Well, it turned out there was an enzyme that Kei's postdoc that was working on that project missed, which was VLCAD deficiency. That is, and the LCAD, the long-chain acyl-collating hydrogenase, is not really a bioenergetic enzyme in people, but in mice it is. So you can knock out one or the other, and you get something that looks a little bit like a fatty acid oxidation disorder, VLCAD deficiency, but not completely. One of the things that it does is it gets cold. You need energy for bioenergetics. If you put a normal mouse in the refrigerator, it just runs around looking for cheese. But if you put a fatty acid oxidation mouse in the refrigerator, it gets cold and it dies if you don't pull it out in time. And so what you can see here is that an untreated wild-type animal just as happy as a clam for 4 hours in our little refrigerator, whereas the VLCAD deficient mice start getting cold. If we don't pull them out by about the time they get to 15 degrees, they die. And so, this animal turns out to be non-recoverable. Typically, we stop here now at 3 hours so that we don't have death as an endpoint. However, if we give them 2 different doses, a small dose and a large dose, by the time we get to the large dose of mRNA therapy, now they can actually survive. They're colder. They're not a normal body temperature, but they're survivable. And we also see here a couple of acyl-carnitines in untreated mice drop. So the pathophysiology of that phenomenon is not 100% clear, and I'm not going to waste time on it right now. But it probably has to do with the ability of the body to make more glucose, because you're providing at least the liver with a fixed fatty acid oxidation cycle, and so there's a possibility that something like this could be useful in animals and ultimately in people. So when we look at the VLCAD mouse, you can see here, that's not like that on my computer. It's missing all of the stuff up here. Well, what I can tell you is it's not hard to imagine. This is a knockout mouse, and this is a wild-type mouse. And when we feed it, or when we inject VLCAD mRNA, you get a lot of it, that curve over here is what happens. These slides got a little bit out of order, and it doesn't get into skeletal muscle or heart. So you can see here, it's in liver, but it doesn't show up in skeletal muscle and heart. So I think this might be an okay drug as a bridge therapy for a baby that's developing, that's fasting intolerant and developing hypoglycemia, and in in conjunction with something like triheptanone, but it's not going to be the answer to everything. And when I say it could be in a combination therapy, it's going to be a bridge therapy, because what do we really want to do? We want to get all of those organs, and we'd like that to be to happen permanently. So to do that, you need gene therapy. And so in collaboration with Korea Pharmaceuticals, we developed a VLCAD expression enzyme, and this just shows that it makes RNA and it makes protein. And when we inject it into VLCAD mice, it makes it's variable, but you get a lot of expression in the liver, which is where the vector goes first. You get a reasonable amount of activity or enzyme in heart, and muscle is a little bit less consistent. But these are males, and in mice, males are more affected than females with fatty acid oxidation disorders. Not true in people. So if you look at the females, which is probably a better model for people, you can see that the skeletal looks better in those individuals. And if we stress those animals, you can see that first of all they don't get cold. Here the red line. And if you look at some other functional studies, so in the liver, as the knockout animals and people for that matter are under stress, you get an accumulation of fat here stained with oil red O, so that the fat globules look red, and here treated with VLCAD gene therapy, and you now get an improvement in the amount of fat that happens. It also improves thermogenesis, and you can see that this is the mRNA slide. Let's just put it in. I guess I grabbed the wrong slide. Sorry about that. It looks okay. It looks like a decent drug, and you can see that here all of the various long chain acylcarnitines decrease. But remember what I said, these animals don't reflect human disease, and if what you want to do, if you want to develop a drug, something that requires you to treat an animal, and then can only do one assay if we have to sacrifice the animal, inordinately expensive. What you would really like to be able to do is give them, is have an animal that manifests all of the features of long-chain fatty acid oxidation disorder. So what we wanted is, instead of an animal that just had a thermogenesis defect, they don't even get hypoglycemic, we wanted one that would get hypoglycemic, have cardiomyopathy, and rhabdomyolysis. So our first thought was, well, maybe we could just knock out the LCAD gene in the VLCAD deficient animals, or breed the two together and come up with a double null. It turns out that's lethal, probably not surprising. So we started on a long-term project where what we did was we made a floxed allele of LCAD in one strain. We're able to breed that to the VLCAD knockout, and then induced the floxed allele with different promoters to come up with different symptoms. And it turns out that if we just induce with a muscle-specific promoter, so now what we have is whole body VLCAD knockout, and just muscle LCAD knockout. What we do, to do that, we get down here to this animal. That took about a little bit over a year for that to happen. When we treat that animal, it looks good. This is the same kind of study that I showed you before, where this animal accumulates fat in the liver. It accumulates fat in the heart. Plus, the heart tissue actually kind of looks normal here. And the heart function is, I don't know if this is going to work. Let's see if it works. Yeah. These are three-dimensional movies. So let's focus on this one. So what you can see is that they actually have a really severe cardiomyopathy starting very, very early on. And not only that, but they start developing, I showed you the steatosis, they start developing fibrosis very early on that looks to be a precursor to the cardiomyopathy. So we've got a good cardiac endpoint, we've got a good liver endpoint. What about muscle? Look at the CP. We run these guys. If you try to run them at the speed of a normal mouse, they just can't do anything. Here's their flat line. They don't go anywhere. They just try to jump off the machine, the treadmill. But if we slow it down, eventually we get something that's pretty reasonable. And they get lactic acidosis with stress running, which is what happens to patients. And more importantly, they get rhabdomyolysis. Look at these numbers, 30,000. That sounds pretty familiar, doesn't it? When patients have an episode of rhabdo, they often go up that high. So, and if we look at a whole bunch more of animals, we can see that well, I'll come back in a second. So now if we treat them and we run them, what happens? Here's a treatment of that animal that now gets rhabdo, has cardiomyopathy, and hypoglycemia with triheptanoin. It really doesn't improve its running at all. So it doesn't appear to be doing what we would like it to do. However, the gene therapy, these guys are able to run a whole lot further. And what you can see here is that their lactic acid does respond with C7, but it responds better with gene therapy. It does improve thermogenesis, so here you can see that now these animals are very, very sensitive to cold. They can't even last 2 hours at cold, and so hitting them with the vector, as I mentioned, females are usually milder. We rescue them completely. Some of the males are still a little bit thermosensitive. So now what we have is a mouse model that completely reproduces the symptoms of patients. We've shown that the gene therapy vector can reverse all of those symptoms, and now we're in the process of going back and comparing all of the various things that we've tested over the years to see if we can see a difference in these animals, because we can follow a measurable marker over time rather than have a once and done kind of experiment. So I mentioned transcriptional activators and the PPAR delta agonist. PPAR delta is a promoter that upregulates most of the enzymes important to energy metabolism. So there's a good reason to believe that it might help in fatty acid oxidation disorders. Even if it doesn't help by inducing a dead enzyme, if you can upregulate to the TCA cycle, which is already active at maximum speed, when you give them a triheptanoin, you can generate some additional reducing equivalents. And if you look here in cells, what you can see is that at least in MCAD, CBT2, and at a medium, moderate dose in LCHAD, you can improve their overall bioenergetic profile as measured by oxygen consumption and have an effect. Unfortunately, this company elected to try, this is Reneo Pharmaceuticals, they elected to try their drug first in respiratory chain deficiencies before I managed to convince them to do it in fatty acid oxidation disorders. The respiratory chain study failed, and they went out of business before we could do this in fatty acid oxidation patients. We did get a phase 1 trial in. It was safe. We saw some effect. And we should, and so I'm talking to other companies hoping to pull it through back into clinical trials. We're getting ready to start a study with Nestle Pharmaceuticals to test ketones, something that many of us have used for decades to try to help our worst patients. And this is just an example of a study from the Netherlands where ketones helped a little bit in some aspects of fatty acid oxidation, but treatment in VLCAD patients, but didn't fix everybody. So this is going to be a 3-hydroxybutyrate. It's going to be a D3-hydroxybutyrate. We'll see what happens. Hopefully that gets started sometime in the next year or so. One of the things that happens when respiratory chain complexes fall apart is that you get reactive oxygen stress. And so going back to that original basic science recognition that this is all a giant complex, we came up with the idea that if that were the case, maybe these patients have increased reactive oxygen stress, and you can see that here that they do. This is a VLCAD deficient cell, and this is a ROS measurement, and you can see it's very, very high. And if you feed them these cells traditional antioxidants, the things you can get in a grocery store or even something that's used in clinical practice like NAC, doesn't do anything to them. But we have access to a series of mitochondrial targeted antioxidants by a very good medicinal chemist in Pittsburgh who is giving these to us. And you can see here, just treating this cell line, there's an effect. So we haven't been able to get a company interested in this yet because we don't have good mouse data. Plus, there's this bias in pharmaceutical world against antioxidants. They think, blueberry juice, pomegranate juice, it's like doesn't work for them. So we're working on it. Alternatives to the triheptanoin. Here I'm not going to be able to show you all of the structures that we're doing, but we have a bunch of different ones. And what you can see here is, once again, looking at reactive oxygen species. No, I'm sorry, this is this is reactive oxygen. I put this slide in. If we treat cells from a variety of different angels are coming down. They're going to fund all my studies. Isn't that wonderful? You get a decreasing reactive oxygen with both C7 and I'll be done in 2 minutes. Don't worry. And some of these, unless the bells go off again, in which case I get completely derailed. With some of these next generation anapyloric agents, and these have the benefit, they're different compounds. You don't get some of the GI side effects that you can see with MCT oil or triheptanoin. And I won't mention, I won't show you anything here, but just in case you're interested, they also work in propionic acidemia and methylonic acidemia because they don't go through succinate. Well, they don't go through propionate. You can make succinate by a different pathway. I mentioned cardiolipin binding peptides, L-mepritide. If we treat a trifunctional protein deficient animal, it gets cold. When we treat it with L-mepritide, it gets better. So now we're going to move very, very quickly to finish up with MCAD deficiency. Remember I told you about chaperonins. Well, it turns out that as I said, the best chaperonins are substrate binding. I had reason for other purposes to prove that phenylbutyryl-CoA, the activated phenylbutyrate that's used in urea cycle defects, is chain shortened by MCAD to make phenylacetyl-CoA the active molecule. So if we treated MCAD cells with phenylbutyrate, and we were able to show that the MCAD activity increased as long as they had a copy of the common mutation, which is a folding mutation. We showed long ago when I was a fellow that if you could stabilize it, it was active, which we did. So we treated 4 patients with phenylbutyrate, and as you can see here, they all started with elevated compounds of one sort or the other, and they all normalized with treatment. One patient didn't have elevated compounds, and so they couldn't do that. Now, So we have a clinical trial going with phenylbutyrate to treat MCAD deficiency. We also have a second trial going with an anaclerotic agent in MCAD deficiency, and it turns out, and here's the surprise, it's triheptanoin, it's C7. If you look at the package insert for triheptanoin, it says contraindicated in MCAD deficiency. Why is that? Because C8 is toxic. Remember, I said I've been around a long time. When MCAD deficiency was first identified, people said, well, I wonder what happens if we give them C8. Well, what happens is they die. And a patient did back in the early days. And so we were looking for, we were coming up with a lot of these anaclerotic agents. We had one that we thought might work in MCAD. We wanted to use something that we knew didn't work, and we had some triheptanoin around. So we gave the mice triheptanoin, and they thrived. You can see here that here's that stress, liver with fat, and when we treated them with a dose that was equivalent to about 0.5 gram per kilo of triheptanoin, they got completely better. Why is that? Well, it turns out that SCAD, the next in the pathway, is active against C7, but not at all against C8. So C8 is toxic, C7 SCAD takes over. And so we now have a clinical trial going, sponsored by Ultragenics, and I didn't mention that the phenylbutyrate trial is being sponsored by Zevra, so they're here to talk to them. And this is being sponsored by Ultragenics, who of course makes triheptanoin. So with that, I will just summarize by saying that by understanding the pathophysiology, we can shortcut the process to making new drugs. There are going to be some new options coming forward, and I hope the next 5 to 10 years for long-chain fatty acid oxidation disorders. And we'll even have some drugs out there for MCAD deficiency that can be used. I view those as something that can be useful for 2, 3, 4, 5 years when the kids are at maximum risk for hypoglycemia, and maybe as an as-needed drug later on. We'll see. So with that, I just say thanks, and here's my lab group on the left, and your left, and my clinical research group on the right, and I'm happy to take some questions.